a fascinating preprint manuscript from the Huang group at UCF. So, inspired by the image synthesizing DALL-E, this report describes the development of a transformer called Cell E that produces a two-dimensional probability density map of protein localization when prompted with a protein sequence and an image of a cell. Unlike previous computational tools that produce discrete one-dimensional predictions of subcellular localizations, the probability density map generated in this method accounts for proteins with multiple subcellular localizations. Since I only have 3 minutes, I'll skip over most of the technical details of the transformer structure and other deep learning stuff, but if these topics are interesting for you, I highly encourage you to check out other video paper reviews, especially for explanations of the development of vector quantized generative adversarial networks, or VQ-GANs. Developed by Esser, Rombach, and Omer, VQ-GANs were implemented in this paper to generate these images. But how does Cell E produce these maps with protein localization probabilities? So, starting with the input sequence, protein sequence, the model uses a previously published pre-trained BERT embedding model to represent the amino acid sequences as a list of vectors, or a matrix. In this model, a maximum sequence length of 1,000 amino acids results in a matrix of 1001 by 768. The rest of the embedding matrix, all with the same number of rows, consists of image embeddings produced by VQ-GANs trained on either images of nuclei or images of protein localization from the Open Cell database. As you can see here, all three of these embeddings are concatenated and used as the input for the Cell E transformer, which is tasked with reproducing the list of tokens generated by these three embeddings. So with the remaining time I have, I'd like to focus on two really interesting demonstrations. So here in the first one involves predicting the location of various GFP-fused proteins. So here in the first two panels, Cell E predicts the native GFP and GFP fused to a random sequence will be localized to the cytosol or secreted to the extracellular space. And when fused to the nucleoplasmin and c-Myc nuclear localization signal sequences, GFP is predicted to localize to nucleus. And this is really interesting because GFP is not natively expressed in humans, yet the GFP sequences fused to nuclear localization signals were correctly predicted to localize in the nucleus. So in the second example, while validating their model against an experimental dataset, they determined a new sequence that could potentially function as a nuclear localization signal due to its abnormally high attention weights from the Cell E transformer. Attention in this case is visualized on the full sequence here in the upper left, with darker shades of red corresponding to higher attention weights. The sequences boxed in blue are previously characterized NLS signals, and the sequences tested in the second row are underlined with different colors. It's kind of difficult to make up, but they should be all there. Unlike the first and third panel sequences with high attention sequences, the second panel sequences was highly localized to the nucleus and had high attention weighting. I should say highly predicted. As expected, inputting the fourth panel's low attention sequence did not result in predicted nuclear localization. It's interesting to note, though, that a significant portion of known nuclear localization signals were not given high attention weights. Otherwise, that's a high-level overview of this novel approach to the problem of predicting subcellular localization. Thanks for watching.